Thank you, Julie. So to start, I'll introduce the region that I'm focusing on, which much of Florida falls within. And this is the Coastal Plain Floristic Province, which is number 36 on that map there. So this is a part of a physiographic region within the US that's defined by a lot of its shared geology of low elevation and a lot of its shared biogeographic history. There are up to around 5,500 different described plant taxa within this region, a very large portion of them coming from Florida. And 30%, around 30% of those are considered endemic to the region. And so for these reasons, for the this sort of shared geography and history, the number of species, and the high proportion of endemism, this is one of the areas within the globe that's has been proposed and selected as one of these global biodiversity hotspots to spend additional, conservation efforts towards. So I want to kind of talk about, as Julie segued into this, some of these ideas of scale, both in some of the realities of dealing with larger scales, and some of the methods that you can approaches that you can take, and when you're working off a larger scale, and then also getting in after that into how scale does, the role of scale in your analyses and your results. And just to quickly kind of show how we went about building our region, our spatial region. This was based off of, we did this based off of EPA ecoregions, the level 3 ecoregions that we pasted together to form this broader region of this defined coastal plain floristic province. And there were some decisions made in how to delimit that. And so there's always kind of some role of decisions going into delimitation, and there are different ways of doing that and choosing your range. But this is how we chose what our geographic extent was. And you can go on to the next. Thank you. And I don't want to dwell too much on this point, but I think it is an important thing to bring up, because it's was definitely one of the largest portions of the time that I spent and putting and working on this. And it's definitely an important thing that a lot of people, whenever you're dealing with broader geographic extent, especially if it extends into multiple states, not only are you dealing with more and more species, but you're also dealing with more concepts of what those species are. And you're dealing with more, flora. It's like each state is going to have their own flora or sets of flora that may disagree over what different species are. And this becomes important because depending on, we're drawing this data, a lot of this data from different data repositories, which I'll point out in a minute. And so what the data that goes into those repositories might not always match some of the concepts of species that you're dealing with. And so just as an example, I was following mostly one flora, which was Alan Weekly's flora of the southeastern US, and in that there are around 5,700, or you can go back, 5,700 native taxa to this area, but going through this, we reduced down to 57 or 4,700 for our purposes of the study of taxa that we're considering and including. And so I'll run through some of this kind of pipeline of putting all this spatial data together. And I won't talk about the phylogenetic aspect. It is, I think it was mentioned, it's a little more straightforward than this, but it's also a big aspect to deal with as well. But I won't go into that today. I'll focus on building all this spatial data. So on the left, when you start with your your taxa list of all of the taxa that or species that you're considering, and then any synonyms that might be useful when you're trying to get different data, because you might have names that are recently updated or and whatever that might be. And what we did is we took this taxa list and we drew specimen records from these large databases, GBIF, IDIG-Bio, and CERNAC, which is concentrated on the Southeast. And so we pulled based off of those names and the families that these taxa are in, a bunch of species records that are from Herbaria, from museum specimens that are aggregated into these databases. And from those records, we were able to pull out location data. So data that had specific localities and that had like georeferences or longitude and latitude coordinates that we could use. You can go to the next one. Thank you. And now, anytime you're dealing with a lot of data, and this is definitely one of the realities and the complications that come, is that a lot of the data isn't great for a variety of reasons. And so you can see that that map on the left shows one species, and I plotted the different localities that we get from these databases. And you'll notice that there are two that are way off on the West Coast. And there are a number of reasons why this might be. Those might have been planted in the garden. It might have been a misidentification or a different species concept. It could have been entered in, the locality, the longitude and latitude could have been entered in incorrectly, or it could just have been just wrong. And so this is something that can have a big effect on the outcome of your data, especially if you're building distribution models. And so, we can use pipelines that try to remove these automatically. And then you can also do some sort of manual checking. And so on the right, I show an example of where we've mapped out all of the records, as well as after this cleaning pipeline. And we can check to see if there were records that were removed that we wanted to be removed, or if it didn't, the automated process didn't work out well. And in some cases, and if you can see on those crosses on the map, those represent localities where we don't have specific information, but because we might have written information of where the locality came from. In some cases, we can produce new georeferences using georeferencing software for species that maybe don't have much data to go off of. All right, you can go to the next. Thank you. And so for this project, we did build distribution models, and we had to automate this process because there were thousands of them. And sorry, one second. And so we did have an automated pipeline that was in R that we used to go through this. And just to quickly show you, we start with those final cleaned coordinates that we have. And then based off of that, we build the area that we want to model onto, which is that polygon that's shown in the pictures. We take the different environmental variables that are being used to build those models, with the climatic or topological and soil variables, and we flip them to that area. We can also automate thinning of some of those coordinates. So in many cases, especially with very common species, there might be certain areas where there's a lot of the coordinates, like maybe they're near a museum or something, and there's a lot of collection or sampling bias that might be happening here. So we can try to account for that. And then it also automates selecting the best performing underlying environmental variables to use, and then fine tuning, testing and fine tuning some of the parameters and selecting the best model. And after that, you can also plot these out and do some manual checking as well to just scan through them and make sure that there aren't any that seem like they're really wrong for some reason. All right, you can go to the next. So here I want to get a little more into what Julie was discussing earlier, and this concept of your geographic extent, that geographic scale, and the role that that plays onto your analyses, because when you have a different geographic extent, you have a different set of species, and so you have a different species pool, and those species pool are going to be those species pool are going to have different branches connecting them, and then you're comparing across a different geographic space. And so I want to show here, and just to keep in mind, a lot of this data that I'm showing is very much in progress, and this is that phylogram. So we're looking at the character divergences, and so when comparing the part of Florida within this larger coastal plain region, and that analysis, and when comparing it to the statewide analysis for phylogenetic diversity, or the sum of all those branches, you see a lot of similarities, and you see a lot of this pattern of that decreasing phylogenetic diversity as you go further south. But there are some differences too, especially up in the Panhandle, and some of that might be have to do with that there are different branches that are represented within this other phylogeny. Some of it might have to do with that you get there's more signal within when you're looking at the Florida state, that you might not be able to see as much of that when you're considering all these species that are also within that coastal plain. And you see some more differences as you go to these randomization tests, where you're looking for significant amounts of high PD or high or low PD. Get advance the slide, please. Thank you. So, for example, in this case, when you're considering the entire geographic extent of the coastal plain, all that region and all the species there, Florida has a very high amount of PD, phylogenetic diversity, relative to the rest of that region, which is not too surprising, because we know that Florida has so much diversity, especially considering this region, and a lot of the species within this region are concentrated in Florida. And when you zone in on just the Florida state and the species there, you start to see some different patterns, and you might notice some different things that aren't really perceptible within the larger extent, but the larger extent is kind of showing you relative to that broader region. Could you go to the next, please? And then this is showing randomization results of relative phylogenetic diversity, which, just as a reminder, is considering the branch lengths. So whereas we're very high in black, that's a lot of long branches, where it's very low in red, that's a lot of short branches. And again, when considering the whole region, Florida harbors a lot of these long branches, a lot of this high amount of deep diversity when considering all of the flora. And when you focus on specifically the state's flora and the state extent, if you see like down in South Florida, those might have a lot of long branches because most of their relatives are in the West Indies. Same for that part in the Panhandle. Whereas when you look at some of these areas within the peninsula, this might represent more of the diversification that's specific to Florida. That's kind of specific to that Florida flora. that you don't see quite as much when you're looking at the whole region, and also just means different things as well. So I think my big point here, and you can go on to the next slide, Israel, is that the results of these analyses are really going to be dependent on what you describe as your geographic extent and the boundaries that you have, that you set in those analyses, because these are, and especially with the randomizations, these are relative. We're looking at relatively, it's relative to what that extent is, to the species that are captured within that region, and then to that species pool that you're considering, because these different species pools are going to be connected by branches in a different way, in different branches. And so it isn't that there is a right or a wrong way of doing this. It's just you are addressing different questions, and so it's important to be kind of explicit with the goals and the questions that you're posing, and that what you're interpreting out of that. You're interpreting something out of whatever species pool of interest and whatever geographic extent of interest, and both of those will show you interesting things. So for example, if you go to the next, just to look at let's see, actually, could you go, let's see, could you go one more, Israel? All right. And yeah, okay. So, and then I just real quick want to end on one final point that's unrelated to this concept of scale, but just to show you of some of the other things that you can introduce into these analyses, including testing various predictor variables on the patterns that you are getting out. So, for example, on the left, we have our phylogenetic diversity output, and you'll notice in that Mississippi River Valley, there's very low phylogenetic diversity, and we can relate this to different sorts of underlying processes. For example, agriculture. This is a lot of stuff that Matt was getting into. We can also, this is a region that has a lot of fire, and where fire is very important for a lot of these communities. So we can relate that to probabilities of fire. And this is also a region where there's community types, a lot of community types that are dependent on like a keystone species, the longleaf pine in this example. And we can relate some diversity patterns to distribution of a keystone species, for example. Just as another thing to kind of end on for all these different ways we could use some of these analyses. Okay. So so we can spend a few more minutes if anyone has questions specific to this section. There are a few discussion questions here that we can go on to, and then we can also end by having a broader sort of panel discussion amongst all of us. So are there any things that coming out of this that anyone has any questions about or any comments on?
I just would re-emphasize how important the taxonomic cleaning aspect of this is. You're absolutely right. That's like one of the biggest factors. It's huge. It's huge. It works. It does also apply to the getting the molecular data together. But I don't know if all of our listeners realize just how many taxonomic conflicts there are, and some of them are very hard to resolve. Some of them are not clean synonymy. Some of them are not what's called homotypic synonymy, that's the easiest, where the synonym is just exactly the same taxon with a different name. Most of the synonyms are not synonyms at all in the sense of the common language. They're partial synonyms. So one taxon name is equivalent to part of another one. And that is just, it's nerve-wracking for this. So we could have a whole separate session on that, but there are a lot of people working on it, and you just have to do the best you can.
And I think that also highlights the importance of using spatial phylogenetics, because there's nothing that binds a random researcher in using species for the terminals. We can limit the terminals to genus level, or go or even below that, and so that gives the opportunity of asking different questions and not being limited by taxonomic information or naming of plants and animals. Which I think is one of the strengths of this spatial phylogenetics. And not to mention the fact that we can even use it for microorganisms and for other taxa for which there's even really challenging to have any specific names for those.
Yes, that's a very good point. And we should be clear that some of the published analyses are not at the species level, they're at a higher clade level. Just because sometimes many of those really tricky synonymy problems and misidentification problems go away if you go to a slightly higher level. So it may not be ideal conceptually, but sometimes it actually, it's cleaner.
Yeah, I think another point along those lines is that when we're talking about phylogenies, we're often talking about groups with shared ancestries, and in many cases there are there are species complexes that might consist of multiple different named species that that haven't completely diverged from each other yet, where there's either a lot of hybridization or one is kind of an offshoot of a broader one, and some of that might be might change some of the analyses a bit if you're making a phylogenetic tree that's showing that they are diverged when they're not. So you may, even that.
That's a great point. In fact, that's another place where you'd be better off taking that whole complex and treating it as an OTU. Matt had OTU in his slides and didn't want to go into the technicalities of explaining it, but for the California data set, the 1,000 and some OTUs that we used were somewhere between the species and genus level. There's 5,000 and some named species and 7,000 and some named terminal taxa. We had just over 1,000 OTUs that represented all of those. And they represented them by using little terminal lineages that there was good evidence for. In some cases, they don't have to have a name. As Israel said, the terminal taxa don't actually have to be a named taxon, as long as you can, justify them, group them. This way it works great. Any of the participants that are using microbial data, it works great, even if you don't have named taxa. All you have to know is the phylogeny and then the corresponding distribution of whatever it is that's at the tips of it.
So let's, I know in this congress that we're doing right now, there are at least 3 or 4 different symposia that have been plenary sessions being organized about the America the Beautiful, which is also called the 30 by 30 initiative. Just wanted to throw it up to the panelists, so all of our presenters, including myself, how spatial phylogenetics can really feed into this initiative, and how spatial phylogenetics can be a very important tool that can be used in making decisions on new reserves. And I know Matt mentioned a little bit about this earlier in his talk, but I think this is a good opportunity to emphasize more on this as well. So do anybody wants to?
Well, I mean, I explained that I've really tried in the California state discussions to get this in there. And most people just look at me and go, and then they just go on. Most of the process in California is, maybe I'm being cynical, but just seems to be driven by things other than rigorous data analysis of where actually would be the best, the best next priorities for biodiversity conservation. You just instantly get into other things that are more political. I don't know, Matt, have you ever actually tried to explain this to a group of policy people that are actually thinking about conservation prioritization? It's a little bit frustrating.
Yeah, I mean, it takes a different approach than talking with other academics, but we've engaged with the California Department of Fish and Wildlife over our results and data and models for this project. They're the ones who are contributing the sort of biodiversity areas of conservation emphasis for California's in-state 30 by 30 initiative. So, they definitely have an interest, but they're also weighing a lot of other important real world considerations. I mean, politics and policy is obviously as important as the science in terms of getting things done. So a big challenge is the communication, and then just this whole field I think is in a pretty early stage to be really taken up broadly, particularly at the national level. I mean, California is kind of on the cutting edge in terms of the scientific engagement of a lot of policymakers and just the quality and quantity of biodiversity data that we have. So I think folks are starting to get interested in that context, but it's just slower at a national level for sure.
Well, even at the state level, that conversation you mentioned that we had with Fish and Wildlife and Nature Conservancy, they wanted our species models. That's the main thing they did actually use from our data are the species models, but try as hard as we did, we couldn't get them to use any of the phylogenetic measures. So at least with the California 30 by 30, it's all based on species, albeit they were very interested in our species models. But,
yeah, I mean, it's a slow process. I think you start by just engaging people and getting them thinking about this new concept of biodiversity, and it takes repeated exposure at multiple levels for people to start moving in that direction. So, it's a long game. It's certainly not going to turn things around to a completely different definition of biodiversity in the course of just a couple of conversations, but I think it's still really important to engage and broaden the perspective beyond just counting species to thinking about what species are where and
Great.
On that road.
Yeah, great conversation. And I think we have a question from Julie just to get the conversation going as well. Tal, you want to read that? You want to coordinate that?
Or I can, if I can. I'll go ahead and ask the question. So yeah, so I guess one of the things I just kind of want to really talk about is taxon sampling, and part of the reason that I'm asking this question is because we're doing a project where we're sequencing a bunch of extinct species, right? And if we're putting those extinct species on the tree and then demonstrating that whole clades have gone extinct, right? And so that's massive areas of phylogenetic diversity that are not being represented at all if we're not including extinct species. And particularly as you've had, humans move into areas, there are certain, particularly vertebrates that have been more prone to extinction, right? So you can imagine if you're using everything in a big tree that there are whole clades that have gone extinct that now aren't represented at all, but you might have one remaining vertebrate there, right? Which then like is representing an entire clade that is now gone. So you know, I just kind of want to think about not just extinct species, but also taxon sampling in general and how that might affect some of our metrics and how we see this moving forward.
Yeah, totally. I mean, we know even with, say, the California taxa, the fossil record shows that there's a lot of other taxa that are gone. You take our sequoia dendron, you know, kind of poster child. There used to be a lot of those. Well, I mean, the trouble with those is two, it's great to add the fossils if you can. The trouble is two. One is being able to add them, having enough data, either molecular or morphological, to add them. And the other is their distribution, getting those terminal distributions in. But I think if you had them, you would add them. Yeah. In fact, you know, we had kind of talked about doing that in that grant proposal that didn't get funded, of actually looking at changes in PD and PE and so forth over time. If you had enough of that kind of information, you could actually see what the trends have been, which would be so cool.
Yeah, exactly.
And then, of course, you could go into the future too. Another advantage of the niche modeling is that you can project it forward, and we can look at least estimates of what are going to happen to the patterns, you know, 50 years and 100 years from now. So I think that temporal aspects would be really cool. I don't know how many taxa we can add, but, you know, the general taxon sampling, you know, is an issue. But I think we solve it kind of like you do in any science. No science really studies everything it could. Like astronomers don't have to look at every star in the universe before they do any studies. So, you know, what affects you is not limited sampling or less than the whole universe, but it's biased sampling. So people always ask me this because almost nowhere in the world do we know what all the taxa are that are there now. And of course, we don't know the extinct ones. So we're always dealing with a sample. And how do we be sure that the sample we have is at least, you know, representative of that?
Representative. Yeah, that's the key, right?
Yeah. So
we do have a question in the chat, asking how can we advance SDN distribution models, I guess, via phylogenetic signal, especially to account for biotic interaction and co-occurrence patterns due to shared environmental space.
Man, I think that's great.
Yeah, I think there's a lot of work being done really kind of at the cutting edge of niche modeling right now that's addressing that very question. You know, it's sort of a mix of some of these spatial phylogenetic methods with imputing niches, you know, imputed trait based niche models. Joint SDMs are sort of one flavor of that look at species interaction patterns. You know, there's so many possibilities that start to open up as we build these large multi-species occurrence and phylogenetic data sets. And I think that's an exciting area. I don't know if other folks have specific experience with that.
Yeah, and also to add to that, thanks, Mark, and thanks, Amin, for that question. So there's also this other field of what we call niche differentiation metrics that have been developed, in which you can, if you are interested in looking into phylogenetic signal of two taxa that perhaps maybe they have a shared history, or perhaps how does evolutionary divergence or diversification that resulted in those two taxa, how is it associated with divergence of their niches? You can model their niches and then see whether there's any overlap or there's any differences or significant differences between the niches, environmental niches of those two taxa or vice versa, or in other ways, and see how perhaps maybe allopatric speciation is responsible for the divergence, and you can use those information and compare them with phylogenetic information to be able to infer what are the mechanisms or processes that are responsible for divergence or diversification of those two staggers. So there are different ways, just like Matt mentioned, it depends on the question you are asking and what are the appropriate methods that go with those questions. And I think there is a comment there that that joint SDMs model species association, not really biotic interaction. So there, yeah, and I think there are different. Matt probably also maybe know more than I do on these, but yeah, there are. So there are different ways of doing the joint models. You can do joint models in which the presence or absence of those data of those taxa are considered as response variables. But there's another way in which you can develop a matrix of presence and absence across the different taxa and use them as as explanatory variable. And so in that way, you are looking into biotic interaction such that those areas that have this taxa, is it the same? Do they also co-occur? What is the level of, yeah, we call it co-occurrence matrix, where you generate a co-occurrence matrix and use it as an explanatory variable compared to when you are using the presence or absence of those taxa as response variables. So there are different ways of being able to model that, yeah. And we can talk about that later on, particularly given the uniqueness of those. Matt, do you want to add to that, or does that resonate what you're saying?
Yeah, that resonates. I mean, I think the basic answer is that, you know, with SDMs, we mostly can't go beyond association. SDMs are an associational tool in general. There are certainly other sorts of ecological analyses one can do to look at interactions, but ultimately SDMs are correlative. So association is the best we can hope for.
Yeah, it'd be really neat to start really thinking about how we can add known associations into these models, right? How they are interacting, but we're not quite there yet, just with basic SDMs, certainly. But I mean, that's where it has to be going. You know, we really do need to start adding in these biotic layers to start understanding these things.
I sort of feel like we should be careful with that, though, because ideally, you would want that to come out of the analysis rather than be you know, input. If I had my druthers, it would be that the distribution patterns are devoid of that, are just the best estimate of distributions per se. And then we would get association information out of our analysis.
Yeah, but correlation doesn't necessarily mean association, you know, so that can be really tricky to make those, you know, it you know, and if you don't have enough data, even though you know two species are interacting, you might not see, you know, without adding that interaction component, you might not actually get the overlap. So yeah, it's tricky though. It's tricky though, Brent. It would be cool if we could start identifying new associations we weren't aware of potentially, right? Because of some overlapping ranges. But yeah.
Great discussions. And at this very point, we want to wrap up this session. It's 3:30 o'clock, and so as to respect the time and so that people can move to other sessions. So we want to really appreciate all the people who have attended this session. I think the internet may have contributed to there were some ebbs and flows in the number of participants along the last 2 hours, but we really want to appreciate everyone. And as this session is being recorded, if you had the opportunity of watching it later on, we also appreciate your time doing so. And we welcome your questions, we welcome your suggestions, we welcome collaborations as well. If you have really cool areas you want to work on and you have enough data, please feel free to reach out to us, and we'll be very much delighted to elucidate biodiversity patterns in those areas. and also to train people and teach people on how to use the different software that have been developed to implement this method. And so we really want to thank everyone. We thank the North American Conservation Congress for Conservation Biology that allow us to give this symposium during the Congress. And thank you so much for your time, and have a great rest of, enjoy the rest of the conference.